this talk was going to go through the population and conservation status of Texas pocket gophers via population genomic tools. So just a little bit more about my interests. I'm interested in small or isolated populations and what sorts of biological characteristics predict their persistence. And we can get some of these characteristics by looking at population genetics, species distributions, and just overall interspecific diversity. Another part of my dissertation I work on is looking at the population genetics of this guy, the Texas kangaroo rat, that is found right now only in Texas. But that's not what this talk is going to be about. So as we've all learned from intro evolution or even in intro biology, the biological species concept, one of many species concepts, is this group of actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. There are limitations to this definition, and as you can see, there's no mention of morphotype or phenotype in this definition. However, the definition of subspecies, these are geographically disparate populations that differ phenotypically. So in this definition, there is mention of morphology. So you can see right off the bat how the species and subspecies can get into these gray areas. Difference in morphotype or morphology suggests a difference in genotype, and within subspecies, there's expected to be gene flow. So if there wasn't gene flow, then they would be species, right? Subspecies are important in a conservation context. They represent distinct evolutionary lineages. There's certain environmental and ecological pressures that they adapt to, and they could, become their own species at some point in time. The Endangered Species Act provides protection for subspecies, and management agencies can declare protections for part of a species range based on subspecies. This figure here is just showing species distribution models and some structure pie charts on Gunnison's prairie dog, where the montane or the mountainous portion of their range is under protection. But there is kind of another side to this whole subspecies endeavor. This is the mammalian species, just a screenshot from the mammalian species paper for Thermometers body, one of these pocket gophers I'll get into here in a bit. As you can see in the map in the middle, there's almost 200 subspecies described, and on the sides is just different, you don't have to try to read that, but there's just many of the different subspecies that have been named. Imagine trying to study all the subspecies for a test. It would get confusing very quickly. So unresolved taxonomy, which if you have over 200 subspecies, it's going to happen, can be the result of populations experiencing genetic divergence at present, just insufficient data or data that's not up to par, or inconsistently applied criteria using different methodologies across different researchers. And one such group, as Jonah mentioned earlier, whose taxonomy is in constant flux, are the pocket gophers of the family Geomidae. And just highlighted here, again, from that mammalian species, almost 50% of the 195 formally recognized subspecies are from Baja California, Arizona, and Nevada. Again, you don't have to try to read this small print. It's just for visualization purposes. So these are the major phylogenetic or taxonomic changes to the 3 genera of pocket gophers in Texas, just in Texas, since 1850 to the early aughts. As you can see, at least for Thermometers, there was a lot of phylogenetic revisions happening, and a couple also happened for Geomes and Kyogeomes. So it's time for a new update, and this is what our project hopes to add to. So why are we studying pocket gophers in Texas? Other than getting to go to some really cool places in Texas, such as Alpine and Fort Davis, couple of things. Pocket gophers, if you're unfamiliar with them, these are subterranean rodents. They're physiological marvels. One thing they can, some species can do is they can turn around 180 degrees within their own skin in a burrow, so they can turn around and go wherever they need to go, and they can push tons of dirt over their lifetime. With pocket gophers, there are 6 genera, over 30 species, and hundreds of subspecies, as you saw in an earlier slide. They're mutually exclusive in distribution on the landscape, typically, and I will get back to that point later on in the talk. And given their solitary lifestyle and strict habitat requirements, these populations tend to be pretty disjunct. So why are we studying pocket gophers in Texas? So I've heard many times, I'm not from Texas, but I've heard that everything's bigger in Texas. So why Texas? It's big. And there's a pretty established pattern that the greater the area, the more species you're going to encounter. Okay. And also being big, you have a lot of ecoregions. There's just a figure of all the different level 4 ecoregions with each a different color for a different ecoregion. So a lot of heterogeneous habitat, you're going to have a lot of different species. And 3 of the 6 genera of pocket gophers that exist, 3 of them are in Texas. And within Texas alone, there are 11 species and almost 30 subspecies. And this map is just showing the type localities of each species and, not each species, but each genus that's in Texas. So the objectives of the project was to assess the population genetic variation of Texas pocket gophers using a technique called RAD-Seq, which I'll get into in a little bit later. Verify these evolutionary histories using phylogenomic methods. Construct species distribution models for each taxa, ultimately with the goal of defining operational taxonomic units. Now you might be wondering, what is an operational taxonomic unit? And again, based on definition you use, it's different for different people. But simply, it can be defined as simply as the group of organisms being studied. They can offer a pragmatic, concrete approach to classifying certain specific groups without that gray area that we mentioned earlier with subspecies, which is necessary for conservation managers looking for strategic guidance, again, with just limited funds. Microbiologists use the term OTU to group bacteria and other microbial organisms based on their sequence similarity, just in case you've heard that in another context. So some of the hypotheses that we generated was that while there is subtle variation, morphological characters that exist within these subspecies, the observed genetic diversity is not significant enough to warrant different management strategies based on subspecies. Reconstruction of the pocket gopher phylogeny will not shake up the tree, basically. Pocket gophers have been studied extensively for many years by many different techniques. I don't think our RAD-Seq data is going to shake up that tree. And these species distribution models will largely recapitulate the known distribution of each group. So jumping right into the field work. So what we did was we traveled to the type locality of each group in Texas, so each species and each subspecies. And the type locality is simply the specific location where that subspecies was described. We would go to those locations or get as close as we can to them, document their presence or absence if they weren't there, collect specimens under standardized protocols, and then we would deposit the tissues at the Natural Science Resource Laboratory at the Museum of Texas Tech in Lubbock. For the lab work, again, we collected the DNA, isolate the DNA using a standard DNAZ protocol. Most samples we used were liver. For some that we could not catch, we got museum loans from, and we used toe clips to get the DNA from that. We sent the isolated DNA off to a sequencing facility in New Jersey for double digest RAD-Seq. And then once we got those data back, we subjected it to some computational analysis using Stacks 2.0, various R packages for population genomics, and RAD-Seq, which is a maximum likelihood algorithm to build phylogenies. So if you're unfamiliar with RAD-Seq, here's just a schematic of how that works. Basically, you have these restriction enzymes, which are the gray Pac-Man looking things up at the top. We use double digest RAD-Seq, meaning we use 2 restriction enzymes. You can use 1, you can use 3, you can use 4, however, whatever you need, but we use double digest RAD-Seq. So let's say the genome is that blue line. The restriction sites are those orange bars, and your restriction enzymes basically cleave to those sites or go to those sites and then cleave the genome in those random locations. You ligate adapters and barcodes onto it, so you'll be able to pair that read back to the individual you got it from. After all that's ligated, pulled together, you can sequence it either single end or paired end sequencing. And then using a variety of different programs out there, you then take those loci and stack them together, and then you enumerate allelic frequency and other population genetic parameters based on that. So zooming in on the RAD sequence stacks, what a stack is basically you have this locus, you have different sites, and as you can see the polymorphism here, in stack 1, at that position it's a C, but for stack 2, at that position it's an A. And what we call those single nucleotide polymorphisms, so SNP, so if you hear SNPs with RAD-Seq, that's basically what that is. And you can use that to differentiate, to basically do those population genetic estimates that we've been doing for years. That's just a rundown on RAD-Seq. So another cool thing that we did with this project, so one way to guide locus identification, locus and SNP identification is to align your reads to a reference genome. Prior to our work, the most closely related genome we could have used was Dipodomys ordi, which is the Ord's kangaroo rat, which the divergence time is like over 30 billion years. So there would be a lot of bias to using that genome to align our reads. So with this, so we were able to sequence and assemble the first 2 pocket gopher genomes, which I think is pretty cool, that we then aligned our RAD-Seq reads to. So for Thermometers body and Geomes bursarius, we do have sequenced and assembled genomes for that. The long read sequencing was performed at Brigham Young University, and the short read sequencing was performed at Novogene. The final assemblies were evaluated for completeness using vertebrate and mammalian protein sets. And then finally, species distribution models. I'm pretty sure many of you are aware of what these are, but basically you have occurrence points or even non-occurrence points, and you have environmental layers. You apply a statistical model to those occurrence points and those layers, and then you get this map of habitat suitability or probability that a species is present in a certain area. So we use Maxent, which is one of the most commonly used algorithms today. It uses the principle of maximum entropy, which basically states that it agrees with what is known without assuming what is not known. And that's just an SDM of Geomes that I'll get into later in the talk. Basically how you read these if you've never seen one before. The warmer colors indicate higher habitat suitability, so like your reds, oranges, yellows. But the cooler colors, especially the blue, indicates low probability of encountering a certain species. Okay, so let me just get into the results. This is a picture I took a couple years ago of the Big Bend region. If you've never gotten down there, I highly recommend. So let's just hop into the genome assembly statistics first. As you can see, both genomes were assembled to pretty good completeness, 92% for both of them. Geomes had a greater PacBio coverage, but Thermometers had a greater Illumina coverage. The assembly length, Geomes had a smaller genome than Mummies, but basically on par with each other. And the GC content down there is about 41% for both, which is right on par with what is known for the GC content in many mammals. So that's comforting. So that was just a brief data on the genome assembly. Now we'll get into a little more of the population the population genetics of each subspecies. So 14 groups were evaluated for Mummies body. You can see all the groups listed here. Loci, how many we were able to generate, their type localities, and the sample sizes we were able to collect either in the field or from museums that passed filtering. And again, you don't have to memorize this table. I'm just showing you guys what groups we actually investigated. So with our genetic data, we ran the data through what's called structure. Basically, it determines a value of K, which is the number of clusters that's best represented by the data. So what you do is you run like K equals 1 through K equals 20, however many you want to do, different replicates of each, and then structure will determine the best possible value of K. If you've never read a structure plot, basically each bar is an individual, and however much is colored of each bar represents the membership coefficient for a certain cluster. So for Mummies body, we tested 14 groups. Structure determined that K equals 5 was the best possible value of K based on genetic similarity. Another way of looking at it is applying, throwing it into a phylogeny, not throwing it, but it's constructing a phylogeny with these groups. And we can see 5 groups here in the phylogeny, and I'm going to just walk you through them. TB ruidosae and TB tularossae, which are the New Mexico Poca gophers that we also sequenced, they belong to their own. North Mountains, I have that in quotes. That's not like a region or anything. I mean, these terms are placeholders, but North Mountains are just basically the mountains that are north of the Davis Mountains. They'll probably change, but those are subspecies Guadalupensis, Pectoralis, Actuosus, Lochagia, and Scotophilus. The Davis Mountains group, that's TB lymphae and TB texensis. Then you have the basins, TB spatiosus and TB pervarius. And then you have the plateaus, so those that in the far east, Limitaris, Confinalis, and Robert Baker. And as you can see on this phylogeny, the branch lengths aren't very long, so there's not a lot of divergence between these groups, and it's a pretty resolved tree. The branches with the squares indicate anything with a support value of less than 0.5. So after you group all those into their operational taxonomic units, basins, Davis Mountains, North Mountains, New Mexico, and plateaus, throw it into a PCA, which is basically a way of visualizing a bunch of data in fewer dimensions. You can see that the New Mexico group kind of separates itself out, while all the ones in Texas kind of overlap. And if you look at PC1, which separates those two, there's less than 10%. That's 10% of the variation. So these groups are very similar genetically. Basically, what this plot is saying. All right, so getting to the species distribution models of the mummies. This isn't a global model like all through the world. Just global meaning all 14 groups being analyzed together. So again, warmer colors indicate greater habitat suitability. Cooler colors, especially blue, indicate areas of poor suitable habitat for the species under question. So the environmental variable of greatest influence were temperature, seasonality, and elevation for the Mummies global model. Then I ran the SDMs again based on their separate OTUs and looked at what the greatest environmental predictor was for each of those. Basins, again, temperature seasonality. Davis Mountains, also temperature seasonality, which could indicate that maybe these two could also be grouped in the similar OTU, but that's just a thought. The New Mexico elevation was the greatest environmental predictor. In the North Mountains, precipitation seasonality was the greatest predictor. And then for the Plateaus group, elevation was the greatest environmental predictor. So just some brief conclusions for the Mummies. Again, 14 species were evaluated, comprising of 84 individuals. Based on structure analysis and phylogenetic analysis, we proposed 5 OTUs. Again, the groups, not going to read through them, but due to the lack of capture success for Spatiosis and Pervarius, which are down there in Presidio and Brewster counties, it is likely that they could belong to the Plateaus OTU, or that even Limitaris could belong to the Basins OTU. Those are one of the groups that, there were intermediate levels of genetic diversity between them. All right, so that's the Mummies. Let's switch gears into Geomes, another pocket gopher genus that's in Texas. This was our largest data set, so I'm just going to run through a small portion of it in interest of time. So 19 groups were evaluated from Geomes. Again, you don't have to read through all that. Sample sizes and average number of loci generated for each group are on this table. Just hopping right into the Geomes phylogeny. Again, filled squares indicate support values less than 0.5. So as you can see, this is a pretty well resolved tree. You get branch lengths of differing sizes, but I want to call your attention to a couple of spots on this phylogeny. So where there are X's, there are some things that require a little more investigation. So at the top, you can see Geomes jugosicularis and Geomes bursarius major are sister. To the left, you can see Geomes bursarius ammophilus and Geomes atwateri are sister to each other. And then over there on the right, you have Geomes bursarius brazensis and Geomes breviceps sagittalis as a sister to each other. So if Geomes bursarius were more closely related, they would be monophyletic, but they are in different places on the tree. So that's something that would require further investigation. So that's what we did. So with Geomes bursarius, 4 groups were analyzed. Of the 3 with type localities in Texas, 2 of them appear to group, at least genetically, with other species. Again, bursarius ammophilus with Geomes atwateri, and Geomes bursarius brazensis with Geomes breviceps sagittalis. Jugosicularis, which is up in Hartley and Dallam counties, up in the Panhandle, they are very similar to genetically to Geomes bursarius major. Global Geomes species distribution model. The variables of greatest influence are elevation and mean temperature of driest quarter. And as you can see, it's a little bit different than it was distribution-wise than the Mummies was. So again, I'm not going to go through all 19 species distribution models for Geomes. That would just take too long. But the environmental variables of greatest influence for Geomes bursarius are mean temperature of driest quarter and elevation. Something interesting to note about the SDM for Geomes jugosicularis is that it kind of overlaps with a similar grouping with bursarius that you can see on the slide here. So the species distribution models and the genetic data, it's possible that jugosicularis at least can belong to the same OTU as Geomes bursarius, even if they are, different species. But more traditional data would be needed. With Geomes at Water-Eye, the counties where they're likely to be found are listed there. Again, no need to memorize them. It's all going to be in a report coming out soon. The genetic evidence, however, suggests that Bursarius ammophilus belongs to the Water-Eye OTU. Similarly, with Geomes breviceps, counties where they're most likely to be found, again, kind of in the eastern portion of the state, and genetic evidence suggests that Bursarius brezensis possibly belongs to that OTU. So just some conclusions on Geomes. Again, that was just a brief rundown on the data that we have for that, because that is our biggest data set. Most groups remain unchanged. Analysis supports 9 OTUs, 1 for each species. Geomes populations in Dallam and Hartley counties are most likely Geomes bursarius. And again, I've probably hammered home about the ammophilus and brezensis at this point, but that's something that should be investigated further. All right, so I know the title just says the Mummies and Geomes, but bonus, we're going to get into Geomes as well. They're the 3rd genera that is found in Texas. And these gopher mounds, they're old gopher mounds found in Big Bend National Park. Just the lighter colored soil, you can see there. So 7 subspecies were evaluated for Geomes. Again, just the 7 species. Their sample size, this is probably our most evenly sampled group. Their type localities and the average number of loci we were able to generate for each. Again, ran it through structure through K equals 1 to K equals 7, and using statistical determination, structure once again determined K equals 5 as the best possible value of K. And with this plot, it's easier to see than it was on the Mummies plot, but I'm just go ahead and label them for you. So Angusticeps and Tamaulipensis are in their own. Clarkii is also in their own group. However, Dalquesti and Perplanus probably form their own OTU, while Lacrimalis and Parviceps form the 5th. So the phylogeny here, again, pretty well supported. That filled square indicates support value of less than 0.8, so very well resolved. And the phylogeny doesn't exactly match up with the structure plot, so there's a little wiggle room in terms of how we could classify the OTUs in this case. For the global Kyogeomes species distribution model, mostly in the Panhandle Plains area, and the environmental variable of greatest influence is annual precipitation. And then the SDMs just by subspecies. So the colored regions show a suitable habitat greater than 60% for the 5 subspecies. Dalquesti, which is in red, Perplanus, which is in green, and those 2 could form a possible OTU. Lacrimalis in blue, Parviceps in orange, those 2 could also form 1 OTU, and then Clarkii in purple. There were insufficient occurrence data for Tamaulipensis and Angusticeps. Tamaulipensis has a locality down in Cameron County, while Angusticeps is found in Maverick County, but you can't build a SDM on one locality. So, brief conclusions on Kyogeomes. Again, 7 subspecies were analyzed. Evidence support 5 clusters. Perplanus and Dalquesti could be combined. Again, that's the red and the green. And then Parviceps and Lacrimalis could also be combined. Orange and the blue on this SDM. All right, so that was a lot to run through. Overall takeaways, from the project. I think probably one of the coolest parts is that the first two genome assemblies for Poca gophers were sequenced and assembled by us. Another pretty cool aspect when I just first started off this project is that we were able to find the Mummies body Olympiae, which was thought to be extinct. So we found that. That was a great day. Multiple lines of evidence support reducing these groups into fewer operational taxonomic units for all three genera. And it's important to note that this is not a taxonomic revision. I'm not suggesting we change names or anything, that more data, more traditional data would be needed to actually revise the taxonomy of these groups. This is just, for management, from a management perspective. Again, we want to, how can we manage many of these groups with limited funds, as Jonah mentioned at the beginning. So one thing that when I've given presentations on Poca gophers for years now, I've always said mutually exclusive on the landscape and never really visualized it until actually I was running the SDMs. And you can actually see that. The SDM on the left is that of Geomes. You can see it's kind of there. That distribution is, eastern Texas, Panhandle, Plains region, and not in the Trans-Pecos region down there in blue. But on the right, you have the Mummies, and that's exactly where you would find the Mummies. So it's just cool to actually, see that in an SDM, that these Poca gophers are mutually exclusive on the landscape. Okay, so a lot of people to acknowledge. Can't go through them all. That add another 5 to 10 minutes to the talk. But I want to provide special thanks to Texas Parks and Wildlife for funding this project, and thanks to Russell Martin, Dewey Stockbridge, and Cody McIntyre for helping us get to some of these locations to capture some of these gophers. The sequencing team at Admira Health, they did a great job with the double digest RAD-Seq. The NSRL in Lubbock, Department of Biological Sciences and at Texas Tech, also Department of Natural Resources Management, National Parks Service, and all current and past members of the Ray, Stevens, and Bradley labs at Texas Tech. Many people helped capture these gophers, go out, dig holes, set traps. So my thanks to everybody who helped in any capacity on this pretty cool project. And I guess at that I will take questions.
Thank you, Michaela. That was fantastic. We do have a couple questions lining up in the Q&A box, and just a reminder, if anyone does have questions, that's where to put them. The first question is by Russell Fowl. He said, for Mummies, are the proposed 5 OTUs supported by all of your methods of data analysis or just structure?
I wouldn't say all of them, but I wouldn't also say just structure. I didn't get into some of the other techniques, but it was not just structure. But that phylogeny also supports K5.
And then Kim Ort asked, from the layer map, Big Bend region is blue, not likely, so why was research conducted there? I think maybe you answered that question when you showed the map of the Mummies being in that area, but maybe do you want to elaborate on that?
Say that again? For the
Big Bend region, it was showing as blue. This was one of the earlier maps you showed, indicating that it was unlikely to find gophers in that region. And so she was asking why was research conducted there?
Right. So I think that first SDM that I did show that the Big Bend region was blue, that was just for Geomes, the Geomes genus.
Right.
But the Mummies is found in the Big Bend region, and Kyogeomes as well. Actually, Kyogeomes is thought to be displacing the Mummies in the Trans-Pecos. So we went to all type localities in Texas, and there were several in the Big Bend region. So yeah, so that first one was just for Geomes, which isn't found in the transpapers. If that makes sense.
And then she followed up with, she said the sample sizes are small. What is the accepted number of samples per for a study for each species?
So in typical population genetics, we want 5 to 10 to 20 samples per group. These are natural populations. In some of those groups, we couldn't get that. But for RAD-Seq, I've read papers where 2 samples were sufficient. So again, I mean, and this is just the nature of studying natural populations. It's you're not always going to get that statistically significant sample size, but we did get something for most of these groups.
Kim responded, "Thank you." Then Sam Kieschnick asked, "Hang on, there was a comment that just came through in the chat. Just to me, it said, okay, I'll get to that one in a minute. But Sam Kieschnick asked, within each genera, would you argue that each of the species is the same ecologically? Geomes breviceps lives pretty much exactly the same way that Geomes atwateri and Geomes bursarius. If so, wouldn't the management for each of these be the same? And before you add in, I will just say, one of the main management things that we do as a management agency is keep an eye on populations that might be at risk, and then consider management actions to try to help recover at-risk populations. And so even if they fill the same ecological role, or if the management techniques would be the same, whether or not we treat them as separate populations will ultimately determine whether or not we engage in conservation action, if that makes sense. So if it's 3 separate populations and we see one declining or having trouble, that would potentially trigger conservation work, whereas if we treated it all as one and one area is declining, well, that might not trigger the same level of conservation work. So that's sort of my answer to that question, but I don't know if you want to elaborate on that, Michaela.
Yeah, I agree with that. My internet kind of spotted out during that question, so I
Okay.
just heard your answer, Jonah.
Right.
So
okay. So do you want me just to reread the question again then?
Yes.
Yeah, so he said, within each genera, could you argue that each of the species is the same ecologically? Geomes breviceps lives pretty much exactly the same way that Geomes atwateri and Geomes bursarius do. If so, wouldn't the management for each species be the same?
Right. I think your answer was depending on whether or not a certain population was, in peril, whether you'd have a different management strategy for that. And I think that's the important part. If they're doing fine, then great. But the fact that we can pinpoint certain populations for targeted management strategies, and that's not my part. That's Jonah and companies. That's what they do. But yeah, I agree with Jonah. With his answer there.
And then there was a question by Grace Darling who asked, what are some of the management implications of the redrawn OTUs?
I would say, so it would be great to be able to manage every population in however specific they need, but there's not enough time, there's not enough money, there's not enough manpower to do that. So basically with the redrawn OTUs, we'll be able to kind of, well, won't say clump because I use that term a lot, but we'll be able to make it easier, I guess. And yeah, if that, I know it's not the greatest answer, but I think it'll be easier to manage if we have, these. It won't be as complex if you have, these smaller populations to try to manage instead of one larger one.
And then
Again,
that's all of the questions that we had from the participants. I have a question for you. I wonder if you could elaborate a little bit on the Limpia detection and were they surrounded by a lot of the Kyogeomes in that area? I know that's been one of the concerns with that species is the encroachment of the Kyogeomes in there. So I'm just curious about if you can elaborate on that detection.
Yeah, so we found that near Fort Davis. I think a lot of our traps we were pulling up Kyogeomes, but we just so happened in this one area that all the traps that we had, well, it was just one in that area, but we went back and there were more the Mummies. So if we went back, because this was several years ago, if we went back, maybe the Mummies would still be there, but it's also a great chance that Kyogeomes would be encountered instead. I know in Alpine, where Spatiosis, the Mummy Spatiosis, was their type locality, but I don't think we found any Mummies Spatiosis in Alpine itself. We've just found Kyogeomes where Spatiosis was described. So, so that's something that's concerning.
Yeah, I also wonder if you can speculate as to if there's any drivers behind this sort of expansion of the Kyogeomes distribution into these other areas.
Right. So Kyogeomes is the biggest of the three genera in Texas, and some hypotheses that have been proposed is that the increasing aridity in that in the Trans-Pecos is kind of. Can you still hear me? My internet.
We can hear you. Yeah.
Yeah. Okay. Yeah. That the increasing aridity is driving that expansion with Kyogeomes, because I mean, if you're a bigger bodied organism, you'd probably be able to, disperse longer distances, be able to dig in places where maybe the smaller Thermometers cannot. I think Thermometers are. I mean, they're found in the rockier regions. So I think with again another hypothesis has been proposed. Roadways, people dumping those soils to make roads has also led to the expansion of Kyogeomes, because they're able to dig in these different types of soils, where Thermometers is just being limited to those rockier soils in the mountains.